Hi everyone, welcome to the second section of Chapter 4. I'm going to put sections 2 and 3 together because there's not so much that I need to show you in this section as, again, a lot of definitions, but also I will be mentioning some ideas to help you remember these concepts and to keep some of them that are very similar, distinct in your memory. So here goes. So here are two items that are often put together, and the main difference is that an observational study doesn't apply a treatment, and an experiment does impose a treatment on the subjects. So observational means that we're not trying to influence, whereas an experiment means we are directly influencing in order to measure their response to our treatment. So please don't get those confused with each other. So cause and effect, which is something we've talked a little bit about, but mostly I put you off and said, hey, we're not going to be able to talk about cause and effect for a little while. Well, we're here. We're here at the beginning of when can we say that A causes B. And experiments are the only, and I like the way they put this, the only source of fully convincing data, because sometimes it's unethical to do an experiment and we still want to discover cause and effect. In the AP curriculum, it is the only way that you're going to be allowed to determine cause and effect or to talk about cause and effect at all. And so, although there is at the very end of this a description of what it would take for an observational study to even begin to try and determine cause and effect, those methods would have to be used in places where it would be unethical to experiment on people, or in whatever it would be that might constitute an unethical experiment, and we'll talk about that too. So make sure that you understand that there is no cause and effect for observational studies in this course especially, but in general in statistics as well. Alrighty. Here is a word that gets confused for something else that will come later. So right now we're just going to establish this definition. Two variables can be confounding when you can't tell from the response in an experiment which of the variables caused that response. And so the book actually gives a couple of good examples. If I can think of one, let's see, sometimes we can think about people with higher incomes have better health or fewer major diseases. And some people might say, okay, does that mean that having money causes better health? Is it the money that causes better health, or is it the fact that the money can purchase health care, which causes better health? Because you can have lots of money and not buy health insurance and not go to the doctor and have checkups. Just the fact that you have money is not the thing that causes the better health. Okay, so there's a simple example that could show you what confounding would be. In some kind of observational study, you would have to think of those kinds of things ahead of time and figure out ways to distinguish between people that have money having better health and people that have money that don't have better health. You have to explore that avenue as well. So you do have to anticipate as much as you can problems, things that might explain your outcomes better than the variable that you're concentrating on. If you don't think about that ahead of time, you may conduct a study and it be a total waste of your time. Okay, so again, this is just a vocabulary page. The treatment that you imply or that you impose on subjects in an experiment is one vocab word. We don't often say experimental units, but it's an important idea. And so if your sample is people, okay, it says, this applies to people very well. The experimental units are the smallest collection of individuals to which treatments are applied. I think that explains itself rather well. So experimental units would be like a subject or a single recipient of an experimental treatment would be an experimental unit. Alrighty, and then subjects, the word subjects, it refers to humans. I think it can also refer to any living being in general. And there's other words you could use. If it's not a living being, You could just call it an experimental unit, I guess. There's probably other words for it, though, that might just be commonly understood. Okay. You will be asked in some questions to to read a description of a study and to tell whether it is well designed or not, and to point out flaws in the design if there are any. Do not assume that there are flaws in a study just because they say to look for them or to describe them. You would be expected to explain yourself, if, no matter what answer you give. But the simplest experiments are the best, where you're not putting so many steps in that it's very difficult to buy an explanation that comes out of an extremely complex experiment. So you define your experimental units really well, and then you describe the treatment that you're going to apply to them. There's a couple other things in between there that we have to talk about after this slide, but then you're going to measure some kind of response that has come about supposedly because of the treatment that you applied to the subjects. All right, and then try not to make it super complex, because the more complex it is, the easier it is to mess up and have invalid results. Also, the idea of confounding comes in here. And so if your response cannot be attributed to your treatment, or could after the fact, you think of something, oh, I didn't think of this, that probably explains this. If you don't do your thinking before you conduct the experiment, you may waste a whole lot of time. Okay, too far. There we go. So doing good experiments requires the idea of the random assignment of a treatment. Okay, so you can't simply apply treatments any way you want. Personal agendas, even unconscious ones, will creep in and cause bias in one direction or the other. And so to avoid that, you would find a way to allow chance to assign treatments to experimental units. Okay, so I'm going to use that phrase, experimental units, even though it feels funny to say. Random assignment of treatments is an absolute requirement for a valid study. So there are ideas behind a valid experimental design that you must include in order for your experiment to be able to yield valuable results. So you have to compare at least 2 different treatments in order to be able to measure responses that you might be able to attribute to a particular cause. You could have a treatment could be you treat group A this way and group B is your control. So lack of a treatment counts as a treatment in one group if you just want to control. Or you could have, let's say you're testing laundry detergents. You could wash a shirt. You could make 3 shirts, the exact same shirts, with the exact same type of stain, and you could wash one with regular laundry detergent and wash another one with that detergent plus stain remover A, and the third one with laundry detergent and stain remover B, and you'd be able to tell how well A and B compare to one another, but you'd also be able to tell how well A and B compare to not using an extra stain remover. So you will have controlled for the conditions to be as close as possible for to the same as for all 3 shirts, but you'd be able to compare the 2 treatments against a control group even. And if you're just interested in seeing how stain remover A compares to stain remover B, you don't have to have a control, but it's always a good idea. You have to have that random assignment to guarantee that you don't have any kind of personal bias creeping in to your experiment. And then if there are variables that could be confounding, you control for them. And we'll talk a little bit after this slide about various ways to do that. But if you know that, you're talking about people who have more money, have better health, and you know that there's something else, like we discussed, that would affect the better health issue, if you don't screen out for that known potential confounding variable, which means you're not controlling for confounding variables, then you will get invalid results. And then finally, the idea of replication, okay? So their definition, it says you use enough experimental units in each group so that any differences in the effects of the various treatments can be distinguished from chance differences between the groups, okay? you don't just have one person in each group, one subject in each group. You have enough of them so that if there is a problem, it can be detected, basically. By the way, any experimental design that you are expected to design and explain in this class, you must do so in enough detail for another person to exactly duplicate the entire process of the study. That means you may have to write paragraphs, okay? But let me save you extra writing. A diagram like this can go a long way to making your answer to the question shorter and quicker to put down on paper and still be thorough. So here we have a sample, a very generic sample of a completely randomized design. All right, so you would draw this picture and then you would describe it in paragraph form. Okay, this particular diagram doesn't show you a control group, but you could put one in there. You could do a third line here and say that you would have a control group that receives no treatment at all, and compare the results. All right, so first you describe the experimental units, whether they're living things or shirts testing laundry or what have you, and then there has to be a random assignment of those things into the two groups. If there is no randomness, your study is invalid. So a random way to assign subjects to groups, and so group 1 gets treatment 1, group 2 gets treatment 2, and then you would compare the whatever variable it is you're measuring after the treatment takes place, you compare those results to one another. So use diagrams to help your explanations of a study design be shorter. Okay, there's always things that can go wrong in an experiment, and you may have heard of the idea of a placebo effect. And that is if you give somebody a fake treatment. So perhaps in a medical study, if you're trying to test the effectiveness of a drug, one group will often be given the drug and another group will be given a fake sugar pill, or if the drug is an injected drug, it could just be saline in the shot they're given, so that perhaps they think they're being given a medicine, they just don't know if they got the fake treatment or not. And that way, any effects that come about when any changes that take place because of a placebo are called the placebo effect. Like when you respond to a fake medicine as if it were a real medicine, that is a psychology concept that is studied very extensively. It's called the placebo effect. So people showing effects of a fake treatment. And that can be a terrible thing that makes your results go completely wrong. Blinding is something you can use to help a study not go wrong. Blinding is when the subjects don't know which treatment they're being given, and double blinding is when you hide from the people in the lab what medicine they're giving to the subjects, as well as the subjects not knowing. So blinding or double blinding we use to keep experiments from going wrong, because we want people's responses to treatment to be natural, especially if it has anything to do with psychology. If people know what they were given, they may over-exaggerate symptoms, that sort of thing. You want to make their behaviors and their outcomes to be as natural as possible, and for the lab workers to not kind of give away the secret of which treatment that the subjects received. Like perhaps in a medical study, that if a worker knows that somebody received the placebo, they might treat them with pity, and then the person guesses they had the placebo, and then it ruins their natural behavior. Is unmeasurable at that point. Okay, so the phrase statistically significant is very fun, especially if you say it 5 times really fast. If you can, I challenge you to do so and let me know how it comes out. It's kind of hard to do 5 times really fast. Statistically significant, and so forth. So when is something statistically significant? This is something we need to pen down. And so the definition of statistically significant, I really like this short, sweet definition here: an observed effect so large that it would rarely occur by chance. Okay, now that has no numbers in it for you to be able to specifically pin down some threshold beyond which some effect is big and can be considered statistically significant. And it is a concept that we definitely need to begin to imply causation, to decide that A causes B. So we will continue to talk about statistical significance. And we will ultimately learn a procedure in inferential statistics called a significance test. So we'll be able to pin that down very specifically. Okay, so blocking is one way to control for variables that you know will affect your outcomes and ruin your study. So I like this example from the book where they were testing out a design for a keyboard that is different from the typical QWERTY keyboard we're accustomed to. And they realized that people that use smartphones would have different outcomes because they're used to typing a lot. And so this may be outdated, but there were plenty of non-smartphones when this study was suggested in the textbook, flip phones and the like, that did not have all of the computer capabilities that our current smartphones have. Those people were not good at QWERTY keyboards and were not typing regularly when there were buttons to push and not just glass on the front of a device that would detect your finger to type, where you actually had to push buttons. Those people would have different responses to different keyboards because maybe they're not as good at typing. And so notice what they're measuring here is typing speed. And so non-smartphone users would naturally have a lower typing speed than smartphone users, perhaps. That's what they decided here anyway. So that's how they split this up. And so among non-smartphone users, this was a block. Among them, they randomly assigned the different keyboards and compared their typing speeds. And among smartphone users, they randomly assigned which keyboard they would use and compared their typing speeds, and then we combine the results. So that's called blocking. I can give you one more example of blocking to control for a variable that might be confounding your explanatory variable that you are testing to see what outcome it produces. Perhaps, I think this one may be talked about in the book, somebody's testing the effectiveness of advertising, and they are advertising a product that they believe men and women respond to differently. And so they might take their volunteers for the study and divide them up into genders. So different genders may view or be affected by an advertisement in different ways. And so they don't want to measure them as a large group mixed together. They measure their genders separately, similar to separating out the smartphone users from the non-smartphone users here. And then they look at the different genders' responses separately and then together at the very end when they go to combine the results. And then they could make statements like, this gender was more affected by this advertisement than that gender. So a block is what we just talked about, and randomized block design is when they then assign the treatments randomly. Okay, and I think the key word here is separately within each block. So we actually are conducting kind of two experiments with two different groups, and then we split them into four because there's two different treatments for each group. So that's something that you have to look out for, different examples that fit that definition. Okay, a matched pairs design is used when you need the subjects to be very identical, to compare two different treatments. So if you were to try and test out some kind of treatment and you wanted to test treatment A on some people and treatment B on some others and try and compare the results, how do you know that the results are actually comparable? So an example in the book is where you measure you and a classmate, you're divided up into pairs in your class, and one of you sits down and the other one stands up, and you each measure your resting heart rates in different positions. And then you compare and see whose is better. But what if you have a completely different body type than your partner, and you're the one sitting and they're the one standing? Are those results really comparable? What if you're different genders? What if one of you has a major health problem and the other doesn't? Would you say that those pairs are matched up very well? Well, I would say no. So the solution in the case of the resting heart rate experiment is to have a single person do both treatments. So for instance, first you have them sit and measure their resting heart rate. and then you wait a little while, and then you have them stand and measure their resting heart rate. And then you can compare, because the person that the experiment was done, the various treatments were done on, is the same person. So you very well compare to yourself, and the differences in their heart rates might have some meaning there. Okay, my favorite matched pairs design came from an SAT question, or not an SAT, an AP exam question, where they were testing out two different brands of boots. And they wanted to test the wear and tear on the boots. And so they gave a pair of brand A boots to someone, and they gave a pair of brand B boots to somebody else, or two different groups, and they wanted to see how the wear and tear on the boots compared. But are there other things that could explain the wear and tear on the boots? Well, what if brand A got put on a big group of people that were all very active, and brand B got put on a group of people that were very inactive? So those results wouldn't be comparable. What do you think is a better way to accomplish a comparison of brand A and brand B? Well, the solution that they came up with was that each person wears one A boot and one B boot. Now, if you're right side dominant, you might have more wear and tear on the right boot than the left. So then they had these people wear them brand A on the left and brand B on the right for a week. And then they had another week where they wore brand B on the left and brand A on the right, so that their dominant side did not skew the results. So that was another way to do a matched pairs design. So watch for matched pairs in this class, because there'll be a slight difference in the type of inference that you do to compare these kind of results. Right now we're just talking about what kind of elements in the design could we choose. So I'm going to actually move on to the very brief section 4.3 here, and talk about the part of it that is in the AP exam. And the ethics part is very, very interesting. And I do mention ethics briefly here and there, but it's not on the AP exam, so we won't spend too much time on it. This slide right here, though, is the most important part of 4.3. This officially tells us when we can determine cause and effect. It also tells us if we can make inferences about the population that we take our samples from. So it's just a little chart here, 2x2 chart, and you have to ask yourself two questions about the study. Were individuals randomly selected, so random samples, and were individuals randomly assigned to groups? And if you can say yes to both of those, then the inference that comes from your study can be made about the whole population, not just about your sample, but about the whole population that sample came from, and you can talk about cause and effect. Remember, these have to be experiments, though, not observational studies. So that's the ideal situation, where you use random assignment for both your sampling and your treatment assignment. Okay. Now, if you do neither, you don't choose your individuals randomly, so maybe you did a voluntary response sample or a convenience sample, and then you personally decided which subjects would get which treatment, then you can't make inference about anything. So those would be poor choices. You would never want to do that. But let's say it's hard to do both and you have to choose. So you could say, okay, no, I'm going to do a convenience sample because I just don't have the money to do the random thing, but I will randomly assign people to treatment groups. Well, then you're not going to be able to make inferences about the whole population since you handpicked people to be in the study, but you could still do cause and effect. And then flip-flop that. Let's say you do have a randomly selected sample, but you handpick them into groups. Then you can make inferences about the whole population, but you would not be able to make inference about cause and effect. So you will have to remember this. This is not on a formula sheet anywhere. That is a key idea in this class. Okay. This is worth talking about for a moment, and then I'll be through. When you do want to say that A causes B, there's plenty of things that you could mess up that would prevent you from being able to make a convincing argument with your study, and other places where you just can't practically do an experiment. Okay. So like the lack of realism in some kind of contrived experiment might prevent your outcome from having any real value. But here's some situations. Does texting while driving increase the risk of having an accident? Well, does that mean we send people out to text while driving and others to not text while driving and see who dies? Well, that's not very realistic, and we would that again. That's an ethical issue, right? How about does going to church regularly help people live longer? So, do we have a group that we tell them they can't go to church for a while? Is that ethical? To say, okay, we're going to make you live a short. I mean, and of course, this would be something like a period of years that it would take to test something like the effect of going to church on your longevity would be a very longitudinal study over time, over years. We're not going to say, hey, you don't go to church because we're doing a study. Or smoking and lung cancer. We're not going to have people smoking cigarettes on purpose just to see if they get cancer. So realism has to play a role in establishing causation, and there are some guidelines for it. So any kind of two-way scatter plot type things you do, you would have to have an extremely strong association in order to establish causation without an experiment. Okay, the association is consistent; it happens every time, not just most of the time. That when you have larger values of explanatory variables, and those larger values always come along with an increase in association and stronger responses to that explanatory variable. Then there's common sense. A cause has to precede an effect. Cause first, then effect. And then the cause is actually a plausible thing. So there's plenty of things where A happens first and then B that wouldn't necessarily be things that would have a plausible arrangement or a strong association. So I think a lot of this is just common sense, but we won't have to worry too much because in the AP curriculum, really, you're only saying that cause and effect comes from experiments, and we will not be throwing scenarios at you where you're trying to establish causation in an observational study only. Okay, so the rest of this very interesting conversation, but not for this video, so perhaps we'll talk about that in a live sync session. Thanks so much for listening.